Good day, everyone. Today we are going to be talking about endocannabinoids, basically endocannabinoid system. And in the course of our outline, we want to actually understand what are endocannabinoids, where are they located, and what are their functions, and also we want to know their biomedical relevance to medical practice. Basically, what are endocannabinoids? Endocannabinoids are molecules that are being derived from phospholipid precursors. Phospholipid precursors. And from the work done so far, they've been proved to be very relevant in pain management, in immunological function, in bone growth, and also various of neurotransmitter modulation. These are the functions of the endocannabinoids. And also is that endocannabinoids could also be exploited for therapeutic goals in oncological process, as it were. So let's go to the topic of the day. Endocannabinoids, we said they are basically derived from the phospholipids precursor. All right. They are basically derived from phospholipids precursor. Phospholipid precursor. And one of the major phospholipid precursor in which endocannabinoid is being derived from is phosphatidylethanolamin, belonging to glycerophospholipids that are being found basically on the surface of the biological membrane tissue we have in our body system generally. And basically, we all know that for a molecule of a ligand to actually carry out its operational function, it must bind to a receptor. So therefore, the endocannabinoids has been discovered to have receptors in our body system where they bind to. And basically, the receptors that are identified with endocannabinoids basically are one. We have endocannabinoid, or what we will call cannabinoid receptor 1, and there are also cannabinoid receptor 2. All these are the cannabinoid receptors in which the endocannabinoid ligands really binds to. And also very important to us is what we call phytocannabinoids. What are phytocannabinoids? Phytocannabinoids are same like cannabinoid molecule that are derived from plant substance. And the major one that we can actually talk about is phytocannabinoids derived from marijuana, or what we call cannabis sativa. And in these phytocannabinoids, the relevant, very active psychoactive substance we have in these phytocannabinoids is what we refer to as detonine tetrahydrocannabinol. Detonine tetrahydrocannabinol. This is one of the phytoactive compound we could find in the so-called cannabis that is very common to everybody. Majority of people do take for recreational purpose. All right. So what is the process via which these endocannabinoids can be synthesized in our body system? One of the major ways in which this endocannabinoid can actually be synthesized in our body system is by a stimuli, chemical stimuli. And this chemical stimuli by way of depolarization, which we are familiar with, whereby a cell has to depolarize and generate action potential that will be sufficient enough to release or cause some signal transduction at the molecular level. So therefore, whenever we have increase in calcium level or activation of metabotropic receptor, all of this will cause the release and synthesis of this endocannabinoid in our body system. The endocannabinoid, for instance, we said they bind to their receptors, and these receptors basically are two: cannabinoid receptor 1, cannabinoid receptor 2. Cannabinoid receptor 1 are majorly found on the central nervous system, the central nervous system of the brain, the CNS. That is where we have much of the cannabinoid receptor 1 located. Cannabinoid receptor 2 has been found predominantly in the immunological cells and also hematopoietic cells. So therefore, if you look at this, because of the location of this endocannabinoid system, they tend to mediate some functional process that are of relevance biomedical activity in our body system. And one of it is that they promote and modulate inflammation process in our body system. They also modulate immunological response in our body system. They also, as I rightly said earlier on, they modulate pain management in our body system. And all of this is also in consonant with areas of the location of this receptor in our body system. And also, these receptors have been found in the reproductive system of the male reproductive organ, such as the testes, where it modulates some activities. One of the important information based on the research work that has been done is that this receptor modulates the production of the male endocrine hormone in the body system. And one of it is that they depress the number of the sperm cells in the spermatozoa, in the sperm cells, and the male reproductive organ generally. They kind of depress. Whenever you see the great expression of these receptors at this region, they tend to modulate the depression of the process of spermatogenesis, thereby making more sperm to be less produced at that area of our body system. So let's continue with the biosynthesis of endocannabinoids that we earlier talked about. Basically, the biosynthesis is based on chemical stimuli. It's based on the chemical stimuli. We said they are based on the chemical stimuli. And such chemical stimuli is elevation in calcium, elevation in calcium, elevation in calcium, and also activation of metabotropic receptor. Metabotropic receptor. Whenever all these two things are being activated, they tend to bring about the synthesis of endocannabinoid system in our endocannabinoid ligands in our body system. So when we're talking grouping endocannabinoids, we could have exogenous endocannabinoids. Those are the ones we take in via the oral cavity. And we could also have endocannabinoids. These ones are the ones the body synthesizes itself. And basically, the major endocannabinoids the body synthesizes that have been worked on and been proposed is One, we have what we call anandamide. Two, we have 2 arachidonide. Arachidonide glycerol. Anandamide is also called arachidonide ethanol, ethanolamide. And if you remember from where when we started this discussion, I earlier said that the endocannabinoid system have been synthesized from the phospholipid precursor. And phosphatidylethanolamine is one of the phospholipid precursors we have at the level of the membrane, via which this endocannabinoid can be derived from. So whenever you have this stimuli being activated based on activation of the metotropic receptors, an increase in calcium based on depolarization of the membrane. Depolarization, you have depolarization here. Depolarization of the membrane will lead to increase in calcium and other process of activation of metabotropic receptor. All this will lead to the biosynthesis of endocannabinoid ligand in our body system. We said it. They perform the function of pain management. They function in immune function. They also regulate the release of hormone, modulator of neurotransmitter and some hormones in our body system. And also is that they do what we call bone growth. They regulate the bone growth system. And also relevance is that they also have the ability to regulate the metabolic process in our body system that you are going to see as we go on in the course of this discussion. So these two endocannabinoids are the major ones that the body has been, the research so far has been able to identify with. We have the anandamide, which is also called arachidonol ethanolamide, and we also have the 2-arachidonol glycerol. How are these things synthesized? They are basically synthesized in our body system via the transfer, via the enzyme. We call it N-acyl transferase enzyme. N-acyl transferase enzyme is basically going to transfer the arachidonic acid moiety on phosphatidylcholine. The arachidonic acid moiety on phosphatidylcholine to the head of ethanolamine. And this is catalyzed via the action of this enzyme, N-acyl transferase enzyme, that requires calcium as its cofactor to actually do the transfer of the arachidonic acid onto the phosphatidylethanolamine. And we've said each other around, all these are phospholipid precursor in our body system, coming from the major lipid content of our body system. So therefore, endocannabinoids are derivatives of the lipids we have in our body system. They are derivative of the phospholipids, basically. So after they've been synthesized, we also, after being transferred, we have the action of the enzyme called NAPE, specific phospholipase. So we have the action of the enzyme called NAPE, specific phospholipase D. Right, this is arachidonide phosphoethanolamide specific phospholipase D enzyme. This enzyme continues the reaction when arachidonic acid has been transferred to phosphatidylcholine to bring about the formation of anandamide. another mand, which is also referred to as arachidonide phosphatidylethanolamine. Understand? Arachidonide, AEA. Understand? Phosphatidylethanolamine. So once this is formed, they are not stored. This is very important. This is very, very important, and it was noting. The endocannabinoids are not stored, unlike the other neurotransmitters we have in the body system that are stored after being synthesized. They are being produced based on demand. And once they are produced based on demand, they are released for their physiological process in our body system. And that's what makes the endocannabinoid molecules being different from that of the general neurotransmitter we have in our body system. Good. All right. So we want to talk about their mechanism of action. They have been released, right? So they need to act, and that is their mechanism of action. Every ligand has its own mechanism of action, every molecule, right? So we want to talk about the mechanism of action of the mechanism of action of endocannabinoid system. Basically, the mechanism of action of endocannabinoid system is via the G protein coupled receptor. The two receptors that have been identified with endocannabinoid system, cannabinoid receptor 1, cannabinoid receptor 2, utilize the G protein, utilize G protein coupled receptor mechanism. And this is a way in which they perform their physiological function in the body system via activation of some signal cascade reaction. And basically, let's just have a drawing or a diagram to actually depict that information. So we have a ligand, that's a ligand which is endocannabinoid ligand, or endocannabinoid ligand. They bind at the surface of the membrane, right? They are binding at the surface of the membrane. Let's say this is our biological membrane. Let's say this is our biological membrane, the lipid bilayer of a biological membrane. So at the surface, we have receptors. The receptors at the surface of the membrane. You have, let's say this is the receptor. At the level of the membrane, we have the biological receptor, which are proteins. Biological receptor at the level of the biological membrane. So the ligand binds here. As the ligand binds here, they activate what we call the G protein coupled receptors that are linked via molecular connection to the molecules of these subunits of the G protein coupled receptors, right? These are transmembrane, 7 transmembrane receptors that are coupled to the protein that are coupled to the receptor. So this is the gamma, the alpha, the subunits of the G protein, right? So by binding What they're going to do, basically, what we understand, they could actually inhibit. What they will do is that they will inhibit the action of the enzyme adenylyl cyclase. And the function of they will inhibit the action of the adenylyl cyclase by binding of the endocannabinoid ligands, to its own receptor, which could be CB1 or CB2, cannabinoid receptor 1, cannabinoid receptor 2. So by binding of this ligand to this cannabinoid receptor, they will cause an activation at the level of the gene protein coupled receptors, which are linked to the biological membrane also through the receptor. So by so doing, they inhibit adenylyl cyclase enzyme. And one of the major functions in our normal physiological and biological understandings is that adenylyl cyclase regulates or catalyzes, the enzyme catalyzes the conversion of ATP to cyclic AMP and the formation of a pyrophosphate at the same time. So this inhibition will downregulate cyclic AMP, cyclic adenosine monophosphate. So by downregulating the adenosine, cyclic adenosine monophosphates, this will lead to other cascade of reaction, and this could also bring about the activation of some signal molecules we call MPK. That's mitogen activated protein kinases. Mitogen activated protein kinases. And that pathway will lead to recruitment of some transcriptional factors, to lead to recruitment of some transcriptional factors, and these transcriptional factors basically will cause some alteration in the gene expression. They will cause some alteration in the gene expression. So basically, that is the major mechanism via which endocannabinoid system works. That's the major mechanism. They inhibit cyclic AMP by inhibiting the downregulate cyclic AMP by inhibiting adenylyl cyclase. And we have said that adenylyl cyclase major function is to catalyze a reaction of ATP to form cyclic AMP and pyrophosphate. And this has been inhibited. So that's the major mechanism of action. So we want to move to the function of endocannabinoid system. What are the functions of the endocannabinoid system? One, because of their location at the region of the central nervous system, because of their location at the region of the central nervous system, the endocannabinoid system has been discovered to modulate neurotransmitter release. They modulate neurotransmitter release. And by way of modulating neurotransmitter release, they control how some neurotransmitters can be released in our body system. And how do they do this modulation? The endocannabinoids basically inhibit the influx of calcium, thereby inhibit calcium voltage ionic channel. And as a result of this will bring about low calcium level, and we know calcium is necessary for the activational process of release of neurotransmitter via depolarization. But by inhibiting and bringing down the calcium level, this will cause a modulation whereby they can regulate the release of neurotransmitter. So basically, this endocannabinoid system inhibits the calcium level in our body system, and this has been achieved through the inhibition of the ionic calcium channel we have in the body system. And to some extent, at the level of the glutamate release, for instance, endocannabinoid system has been discovered to actually inhibit the release of glutamate neurotransmitter. And we know that glutamate is an excitatory neurotransmitter we have in our body system. And once its production becomes so much that they accumulate, they can lead to what we call excitotoxicity. They can lead to excitotoxicity, and by way of excitotoxicity, this will lead us to necrosis of the neurons, neuronal necrosis, and further going into death, and which is a condition called neurodegeneration via accumulation of glutamate receptor. And this is one of the major functions endocannabinoid system has been discovered to do. They do what we call neuroprotection by reducing the amount of glutamate that accumulates at the level of the synaptic membrane. And before I go to the next function, let me quickly say this at the same time, that the normal transmission, synaptic transmission in endocannabinoid system is a bit different from that of the normal neurotransmitter, because what we normally have in the normal neurotransmitter is that the neurotransmitter, after being synthesized, they release via the presynaptic neuron, and they go to bind to the postsynaptic neuron for their target receptor. But this is different in the case of the endocannabinoid system. What happens in the endocannabinoid system is that you have the endocannabinoid ligand synthesized from the postsynaptic neuron, and the flow presynaptic neuron via the transporter, protein transporter. You have this protein transporter that mobilize them from the postsynaptic neuron to bind to a receptor that are located on the presynaptic neuron. And after they have been formed, they've carried out their mechanism of action, they've executed their mechanism of action, they flow back again to the postsynaptic neuron, where we have the enzymes that will biodegrade them after their action. All right, so let's go back quickly to the functions, where we start the functions of endocannabinoid system. The second function of the endocannabinoid system is immunological function. Immunological function. The endocannabinoid system has been recognized to perform a major important role in the immunological function. How they do this is basically they upregulate and activate the action of lymphocytes in the body system, thereby promoting the synthesis of chemokines, which are very important in inflammatory function. And this is also tied to the location of the receptors. And we have said cannabinoid receptor 2 are basically predominantly abundant in the immunological cells. So they perform this function of immunological function by activating some immunological cells such as lymphocytes in the body system. Another function of the endocannabinoid system is that they regulate the metabolic function in our body system. Metabolic function. Metabolic function. And when we mean that the metabolic function, we're just talking about the ability of the endocannabinoid system to regulate the metabolic activities of the food intake. And basically in the body system, we have the region of the brain that is wired up to control the amount of food we take, leading to what we call satiety. Understand? Regulating the amount of food we take. And this is being done by the hypothalamic propiomelanocortin. Understand? Hypothalamic propiomelanocortin. And basically, because of this function, the endocannabinoid system inhibits the release of alpha MSH, which is being released from hypothalamic propiomelanocortin. And by inhibiting the release of alpha MSH, melanocortin stimulating hormone, right? Once this is inhibited, it brings about increase in food demand in the body system, bringing about what we call increased appetite. Understand? They bring about increase because the major function of the alpha MSH, which is being released from the hypothalamic propiomelanocortin, is to inhibit or give the body that signal of feeling that enough food is okay for the body system. But in this case, the endocannabinoid system will bring about what we call inhibition of the alpha MSH, and by so doing, it upregulates the satiety and more demand and craving for food. And this can be seen in the general intake of people. You know, the people that take cannabis sativa, they tend to increase the amount of food they so they tend to increase the quantity of food they want to have because of the action of inhibition of the endocannabinoid system at the level of hypothalamic propiomelanocortin level of our body system. So people taking cannabis, and cannabis sativa help, we get, so they, as a recreational substance of recreation, they tend to have more increase in food intake because of these molecular activities that happens at the level of the brain. That is the metabolic function. Another important function of the endocannabinoid system is that they also regulate bone tissue growth. They regulate bone tissue growth. This has been proposed to happen by way of activating some growth molecules, more growth signal molecules in the bone tissue. And by so doing, the endocannabinoid system can actually bring about increase in the proliferation of bone growth also. And another important function of the endocannabinoid system, is that they regulate gene expression. They regulate gene expression by activation of some transcriptional factors at the molecular level, such as MAPK, which we call nitrogen activated protein kinase. And by so doing, they can alter the gene expression we have at the level of our body system. That's a function of endocannabinoid system. And now we want to talk about the biodegradation of the endocannabinoid system. How are they degraded in the body system after being synthesized, and they've actually also carry out their biological function that they are meant to do in the body system. We have two important enzymes. That are responsible for this very action in the body system. One, we have what we call fatty acid amide hydrolase as one. Two, we have what we call the monoacyl glycerol lipase enzyme. These are the two enzymes that have been identified to actually degrade the endocannabinoid ligands after they've carried out their function in the body system. So, in other words, these two enzymes regulate the activities of the endocannabinoid system in the body system. So, we call fatty acid amide hydrolase. We biodegrade anandamide, damide, while the monoacyl glycerol lipase, we biodegrade to arachidonide glycerol. So these are the two enzymes that play a very significant role in the metabolism of the endocannabinoid system that are being formed after they've carried out their synaptic function and their biomolecular information they need to carry on. After they've done that, these are the two major enzymes: fatty acid amide hydrolase, which catalyze and biodegrades anandamide, and we also have the monoacyl glycerol lipase that also catalyze the biodegradation of arachidonide glycerol. Let's talk basically about the clinical correlates of these two, clinical correlates of the endocannabinoid system. The clinical correlates of the endocannabinoid system. One, they serve an important role in cancer therapeutics. Important role, they play an important role in cancer therapeutics. And one of the major information concerning this is that, for instance, in some cancer cells, it has been discovered that activation of the endocannabinoid system will bring about the activation of what we call ICAM, intracellular they bring about ICAM, intracellular activation of intracellular adhesion molecules, ICAM, intracellular adhesion molecules. And by way of bringing about this, they prevent metastasis of cancer cells in the body system, and also by way of being synthesized in the body system, by way of these endocannabinoid ligands being synthesized in the body system, they also function to protect the neuronal cells, to protect the neuronal cells, and by way of protecting the neuronal cells, they inhibit neurodegeneration. And this can be utilized, this ideology can be utilized in the development of medications and of therapeutic goal that can be used, for instance, in some of the neurological diseases such as Parkinson's disease or Alzheimer's disease. So all of this is playing a very significant role in making sure that our body system is also very important to that signals of the endocannabinoid system. So at this junction, I would like to say thank you very much at this juncture. I want to say thank you very much for listening. See me again when I present other lectures in relevance to biomedicine and medical biochemistry courses. Thank you very much.